Hello, this is our ancestry testing, the S&P of prostate cancer. My name is Javier Gonzalez.
My name is Natalie Hedden.
I'm Jacob Kropp.
I'm Harpreet Kaur.
And I'm Kaylyn Lopez.
I will now be passing it to Kaylyn for the objectives.
The purpose of this project is to analyze and interpret the raw genotype data. We identified at least one human genetic disease that is caused by a single nucleotide polymorphism or SNP. Our analysis is based on the dataset provided by a human ancestry testing company. We researched the single nucleotide polymorphism to determine the disease risk and how the SNP leads to a cellular effect that causes disease. Lastly, our goal is to make a prediction about the potential risk of the anonymous donor. And now I will pass it on to Harpreet.
Single nucleotide polymorphism, or SNP. SNP is the most common type of genetic variation between individuals. SNP is a point mutation and can also be defined as an individual nucleotide-based sequence between two DNA sequences. The figure on the right shows a single base pair difference between two DNA sequences, and this is an example of SNPs. In humans, for a variation to be considered a true SNP, it must occur in at least 1% of the population. It also provides an important source of molecular markers that are useful in genetic mapping. Now, I would pass it on to Javier for the introduction.
For the introduction of prostate cancer, it forms in the male prostate and can be the size of a walnut in younger males or even larger in older males. Most prostate cancers are adenocarcinomas, although some other rare types of cancers can form. Men can be tested for prostate cancer by having the gland cells observed under a microscope, and they can be formed through a single nucleotide polymorphism, otherwise known as SNP. The figure on the right shows the Cleveland Clinic's information on cancer. It shows that 1 in 6 men will be diagnosed with prostate cancer during his lifetime, but most men diagnosed with prostate cancer do not die from it. In fact, around 2.5 million men have been diagnosed with prostate cancer and are still alive today. I'm going to go ahead and pass it off to Natalie for the genetic background.
This specific ancestry test showed that SNP-RS109939994 is the point mutation that causes prostate cancer. SNPs associated with prostate cancer risk are associated with gene expression changes at the mRNA level. These cause the genetic factors associated with prostate cancer risk in both the normal and cancerous prostate tissues. I will now pass it to Harpreet.
Results and methods. The OMIM database was used to search the genetic disease prostate cancer. The OMIM search gave us the RS number for the SNP that enhanced the risk of this disease. The RS number was then put into SNPedia to get more information on the genotypes and phenotypes, and to get the horizontal bar graphs. We were provided with 2 datasets, and those datasets were listed as RS-ID number, chromosome number, nucleotide position on the chromosome, and the genotype. The dataset was of an anonymous donor provided by an unknown ancestry testing company. This dataset was analyzed to assess the risk of developing prostate cancer in the donor based on the genotype. Figure on the right shows the OMIM search of prostate cancer. Now I'll pass it on to Jacob.
Okay, so further looking closer at the prostate cancer, the SNP RS10993994 is associated with the prostate cancer. Values that were greater than 90% were established where inputted SNPs were correlated. Evidence at the 2 loci 512190 through 512380 did find evidence of prostate cancer existing there. The prostate cancer is found on the locus 1 in the 5' untranslated region, and this is just abbreviated for UTR, which is a genomic DNA sequence that's not translated in the RNA sequence. So this basically refers to either side of the 2 sections of the coding sequence that's not being translated. As you can see down in the bottom left corner in figure 1.4, the gene, the RS10993994, is on the 10th chromosome. And the genotype here is reflected by this anonymous donor is the CT genotype. And as you can see onto the right in the bottom right corner in figure 1.5, the risk based on the genotypes varies with the CT. As we can see here that it is 1.5 times more likely to have an increased risk of prostate cancer. I'll now pass it off to Kaylyn to interpret this data.
The SNP RS10993994 is located 2 base pairs upstream of the gene transcription start site. The T allele on the gene is associated with an increased risk of developing prostate cancer because the T allele changes the transcription and splicing factor binding sites. On the right side, figure 1.6 summarizes the risk of developing prostate cancer based on genotype. The risk is described in order of magnitude. It is found that individuals with the CC genotype are at no risk, whereas those with CT or TT genotypes are at an increased risk of developing prostate cancer. Based on this information, we found that the anonymous donor is at an increased risk of prostate cancer because they have the CT genotype. And now I will pass it on to Natalie.
We hypothesized that the male in this specific ancestry test cannot be conclusively determined. The highest tested CT ranged from 48.9% to 49%, which can be seen in figure 1.7 and 1.8. This could be due to multi-ancestry analysis being used to identify the population-specific risk of the SNP. I will now pass it to Jade.
All right, so looking back on our reflection on the SNP of prostate cancer, we did learn that it's the most common type of genetic variation between individuals. It does give this an added advantage in our skill set. In addition to not knowing that SNPs can cause a genetic disease or lead to phenotypic effects, it does provide an important source of molecular markers that are useful in genetic mapping, which does persuade us to stress the importance of human DNA sequencing. The relevance to society and individual loved ones to learn about DNA sequencing and ancestral history can make an immense impact on the diagnosis and the genetic inheritance of how we can treat prostate cancer. The background knowledge we learned through this report proves useful not only in our future applications, but also in advocating for DNA sequencing. I'd like to thank you guys for listening to our presentation today on prostate cancer. We also have on the other slide our resources and our author contributions.